This is part 2 for chapter 10, antimicrobial drugs. And for these slides, we're going to be focused on drug resistance and looking at how drug resistance develops, first of all, so different mechanisms that bacteria have to gain resistance to different types of antimicrobial drugs, and also talk about the concept of natural selection and how that relates to what's happening inside the body when someone takes medication. We will talk a little bit about alternate approaches, and I'll update these slides on Canvas because your book has kind of limited information here, but there are definitely some newer technologies that are being developed that can be used instead of traditional antimicrobial medication that have potential benefit for therapeutic application. And then we'll finish this set of slides, these sets of slides, by looking at how to go about choosing the most appropriate antimicrobial drug. Now, again, this is for those of you who are entering into healthcare profession, this is just things to consider when you're essentially in the same room as your patient and the physician is prescribing antibiotics. Just stuff to keep in mind also as first runners in the field of healthcare that you definitely want to understand how drugs are selected and considerations to make sure there's no contraindication or damage to the host. So let's start with drug resistance. So there are two different ways that drug resistance can develop. This is either through just random spontaneous mutations that occur. We talked about this in chapter 8, that for every replication event, every DNA polymerase from every organism has known rates of mutations, right? Sometimes if these mutations occur in coding regions, it can lead to alterations, for example, of drug targets. So if there's a mutation in one of the folic acid pathway synthesis enzymes for bacteria, and we provided something like trimethoprim that targets that particular enzyme, that enzyme might not have a perfect fit anymore for that drug. And so therefore that spontaneous mutation, although inherently detrimental to the bacteria, can potentially protect it from antimicrobial drugs. The second means by which organisms can gain this drug resistance is through direct gene transfer. We talked about 3 different methods for bacteria to transfer genes in a population. If you recall these ones, right? Direct from one organism to another. Conjugation would be one, using that sex pilus and transferring that F factor. We're going to talk about that one again, but this time we're going to refer to it as an R factor if it contains a drug resistance gene. Transformation, right, through the acquisition of chromosomal fragments or plasmids that are in the environment, or transduction through bacteriophage. So let's talk about these a little bit more detail, right? For spontaneous mutations, we know, if you know anything about, we've talked about mutations already. Mutations are inherently detrimental, right? They change the DNA from the wild type to a mutant version and has potential impact on the function of that gene product, that protein, that enzyme, that regulatory molecule, whatever it might be. So although mutations are usually detrimental, this term here that they can be beneficial, well, this is just in relation to how potentially they could be protective, right? Whoops. So these are 2 different kind of concepts here, and I just mentioned one. So if the mutation occurs in a drug target, then it could confer a change in the sensitivity of the organism to the drug because the drug target is no longer that perfect match anymore. If you remember back, and let's just kind of draw a picture to remind us because it's been a few days since we have looked at chapter 7 and chapter 8. If this is, an enzyme, and remember we kind of draw it this way, and we have this active site, and we know that this enzyme matches perfectly with that substrate. Many of these drugs act as a substrate mimic, right? So they'll have some kind of format that makes them look super similar. And that would be like your drug equals a biological mimic. which also means it's acting, if you remember how enzyme regulation occurs from chapter 7, this would be direct enzyme regulation, and this would be competitive, right? Through competitive inhibition. Other drugs might act allosterically, right? So a drug could be an allosteric regulator. And the way that antimicrobial drugs work would be that regardless of what the mode of interaction is, the concept here is that this is an irreversible docking. So if you remember back when we talked through chapter 7 about competitive inhibition or allosteric regulation, non-competitive inhibition down here, that for the natural way of enzyme regulation, these must be reversible so that when that organism needs that enzyme again, it can remove these inhibitors and allow that enzyme to function. Now for antimicrobial drugs, we don't want the bacteria to be able to remove the inhibitors. So usually these dock and form covalent bonds inside either the active site here or the allosteric site down here so that these become irreversible and therefore the enzyme becomes deactivated. But again, if you remember our chapter 7, we talked about how the substrate and enzyme must have a perfect match here, chemically, sterically, right? They have to have this perfect interaction between the substrate's binding area and the active site on the enzyme. So for the competitive inhibitor, this must also have a really, really tight fit so that it can actually compete against the substrate. Now, if there's a mutation, let's say there's a spontaneous mutation in the DNA, right, of this bacteria, and it conferred a developed, there was a mutational event that was in the active site or somewhere in the formation of the active site here that caused an alteration here, right? So if, let's say, there was this spontaneous mutation that occurred in the active site or nearby, then this mutation might make the active site less able to bind to the substrate. So we know that this could confer loss of function or a less functional enzyme, right? So it's detrimental. We talked about this before, that if you have alteration here to the active site, that it could cause loss of function or lowered function because it doesn't dock as well to its normal substrate. But the benefit of this mutational event is that also it would then confer resistance to the drug, or at least a loss of sensitivity. Right? So a mutation can, even though it inherently could be detrimental to the organism, it also may be have some benefit in at least drug resistance capacity. And then this mutational event could then be propagated in the population to confer that resistance to other organisms in that population if indeed the drug is present and therefore this mutation would be selected for, right? So this is kind of what this is talking about. This is one example of how a spontaneous mutation might confer some type of benefit or resistance to an antimicrobial drug if the drug target is altered. There are other ways that this can happen through various means, and I'll show you that in just a minute. So that would be spontaneous mutations, but the other way that bacteria can transfer resistance or acquire resistance is through the direct transfer of something we call an R factor or resistance factor. Through ideally conjugation would be the primary means by which this entire plasmid that contains a drug resistance factor on it could be transferred from one organism to another. But we mentioned already, we know that transfection and transduction are other means that these genes could be propagated in a population. So these drug resistance factors, these particular plasmids, that's what they are, right? Let's just mention that this means these are plasmids. that contain, excuse me, some kind of drug resistant gene. That contain a drug resistance gene. Right, so some kind of enzyme or a gene that encodes for an enzyme that may deactivate that drug or may prevent its transfer into the cell, or there are many ways that drug resistance can be promoted by different types of genes, by different types of enzymes. So these resistant factors are readily transferred between organisms, and this becomes especially pertinent inside the human host, where we have lots of different organisms in our gut. Pathogens can come in, and that drug resistance factor can be sort of spread around rather quickly. This is just a picture to remind you of these three different mechanisms of gene transfer, and now we're just going to focus on how these would be potentially resistance genes. And so let's talk about what these resistance genes might be. But here again, plasmid transfer through conjugation, transfer of free DNA. This is a chromosomal fragment or a plasmid that is released from that lysed cell that could be taken up by a neighboring cell or through viral delivery. So let's look through what these mechanisms might be, right? So here are some examples. Drug inactivation, we've talked about this one already. And if you recall, one of the means of drug inactivation, we talked about this in the last set of slides, the beta-lactamase. Right, ACE means enzymes. This is an enzyme that degrades the beta-lactam ring on the beta-lactam drugs like penicillin or psyllin derivatives, and can be inactivated, as we've mentioned already before, by clavulanic acid. But this is an enzyme that could be propagated in the population very readily through these resistance factors and provide drug resistance for any of those beta-lactam drugs. Drug permeation. Permeation means to penetrate through, like to get inside, means getting inside the cell. So all drugs that have to go inside the cell, which is almost all of them, must have some way to get in. Remember that cell membrane is actually rather impermeable to most things. And so these drugs must have some type of receptor or some type of transmembrane protein that they can bind to and that allow the passage of those drugs into the cell. So if there's a mutation to that permease enzyme, that transport protein, then you may lose that sensitivity to the drug because drug just can't get into the cell to target its drug target, or to get to its drug target, right? Drug receptors or drug targets, we already mentioned that one before. So mutations can alter the actual drug target itself, the enzyme that's being regulated by that antimicrobial drug, and that would lose sensitivity for that organism. And then again, that spontaneous mutation, even if it occurs in one organism, if that drug is in the population, then that mutation will be selected for, and therefore that mutation essentially can propagate in the population due to that selective pressure. And then some organisms are able to actually flip altogether. So if a drug is targeting one metabolic pathway, then they can upregulate enzymes involved in a parallel pathway so that they can not be resistant anymore. Here's a picture of some of those things, excuse me. I got something in my throat. So here's a normal non-resistant organism where a drug permeates that cell envelope, including the cell membrane. And once it gets inside, it has a drug target that it binds to, and it can inactivate that, that drug target, that enzyme. And recall that most of the classes of drugs that we have are enzyme targeting drugs, right? So cell wall synthesis means the enzymes involved in synthesizing that cell wall. Ribosomal complex, there are many different ways that these drugs can target the ribosomal complex, as we talked about before. So whatever that target might be, the drug has some physical interaction with that target, and it confers inactivity of that metabolic pathway or replication event and then can kill that cell. So here with a resistant cell, there's a whole bunch of variations here that we can talk about. Here's an example of increased elimination. I didn't mention this in our last slide, but drugs that enter in could also be pumped back out again through these efflux pumps. And this does occur in some organisms that have these efflux pumps. Again, that would be from a gene that encodes for that transmembrane protein that can then be passed in the population. Drug inactivating enzymes, as mentioned, this would be like the beta-lactamase or other enzymes that can go and modify, physically bind to and modify the drug so it no longer can target its actual drug target. Alteration in the target molecule. So here, again, an example of like a mutational event that perhaps could then be propagated in the population where the drug just cannot bind to it anymore because of that mutational event. And then here, decreased uptake. So for example, the lipopolysaccharide porine proteins might prevent the entry of many drugs into that gram-negative cells. So they have natural resistance just due to the prevention of uptake, right? Or as mentioned before, that permease, that enzyme transmembrane protein that allows the entry of this drug, if it's mutated, then it can prevent the entry of that drug altogether, right? So these are variations of mechanisms that can confer resistance to different antimicrobial drugs. I'll talk through all of these ones. You can look at those yourself. Okay, so there are other ways naturally that organisms can be resistant. Dormancy, we kind of know about this already, but this is especially true of sporulating bacteria. Right, so if a sporulating bacteria faces an antimicrobial drug, they could immediately begin to sporulate, and those spores are then protected from any of that drug activity. And when the drug is then gone, because the person stops taking it, then the spores can germinate potentially and develop in the absence of the other organisms. There are other forms potentially that bacteria can enter into, so phase shifts, so they shift their metabolic activity essentially to a more dormant form, and they just kind of coast through that that time period and then can reactivate. That's not as common as sporulation. And then the L form, this is a term that we use to talk about cell wall deficient organisms. So they're, as long as they are either adapted to that changing osmolarity, right? So that they have other mechanisms to prevent the lysis in the face of osmotic pressure, then they can exist in this L form, and therefore any obviously cell wall enzyme drugs would have no effect on these types of organisms. This is also true if you, right, if you have a bacteria in an isotonic solution and you throw a cell wall synthesis drug in that solution and you continue to grow the bacteria, they're not going to die because they are not facing any osmotic pressure, even with the absence of the cell wall or the loss of integrity of that cell wall. They won't die because there's no pressure to lyse the cells, right? So consider these things. So let's talk through natural selection here and why drug resistance is such a bad thing, and really more the concept behind what's kind of happening in the body. So natural selection is when the environment changes, and that change selects for particular traits in the population, right? So it's all about adaptation to changing environments. Let's look at this example down here. Let's say that organisms, as they grow, let's just say this is one population, and there was a spontaneous mutation that occurred maybe a while back in some particular drug target, and it just, that mutation, even though it's present and it confers resistance to a particular drug, this drug resistance gene will really not have any reason to propagate in the population, if you want to think about that, if you want to humanize this thing, right? So it might spread naturally just because of the offspring of this guy, or some perhaps a lysed bacterial cell, and this, you know, another neighboring organism picked it up. So in a general population, a spontaneous mutation really will not be spread unless it provides some type of benefit. If it's a drug resistance gene, it's not providing any kind of benefit in the absence of that drug, right? And in fact, these spontaneous mutations that are slightly detrimental to the organism will probably actually be selected out of that population. If there is no benefit it confers and it actually is detrimental, then they're less fit to a standard environment. But nonetheless, let's just say that this drug resistance gene is in just a couple members of this population. Now, when selective pressure is put onto that population, in other words, that drug is applied to the entire population, well, now we have selectivity towards organisms that are better adapted to that new environment, in this case, the presence of the drug. So early exposure to the drug, a whole bunch of the organisms will get wiped out. The organisms are still growing over time, if you recall. So these resistant organisms will not be destroyed and will continue to propagate, right? So late term, you might have a few organisms that are less sensitive due to other factors, right? So they may have resisted that drug or were not impacted by that drug right away. And then you have those those actual ones that are resistant to the drugs that are able now to sort of propagate a little bit more. Once that drug is removed, even though there's no more selective pressure anymore, you have just changed the population genetics during the event, during that selection event, so that even in the absence of that selective pressure, now the gene pool has been completely changed, because the progeny, or this population, will all be the progeny of whatever that, that population that was left behind, right? So this is a perfect example of evolution at work, change in the genetic makeup of a population over time, right? And in this case, it's due to the selective pressure that's put on the drug. Now let's consider this inside the human body. So let's say that this is your gut, and here's my fabulous drawing. Here's you, and this is your gut, right? Whatever. So this is your gut, and inside your gut, you got your normal flora, and right, most of them don't have inherent antimicrobial resistance factors, but there actually are some. In fact, my lab, the lab that I belong to at UC Davis, we've just recently published, not me, but some members of the lab have recently published a couple of papers looking exactly at this, at antimicrobial drug resistance, or antimicrobial resistance genes that are found in healthy individuals in different populations, right, based on the microbiota, and really kind of looking at inherent resistance from different organisms. And unfortunately, pretty much everyone has drug resistant genes in their microbiota now, even if you've never taken a course of antibiotics in your life. Even brand new babies that come out of their mama, and the pregnant woman never had antimicrobial drugs, and the baby was never exposed to them during delivery, there will still be some antimicrobial resistance genes found even in that infant, right? So it's just in the population. Over time, we have evolved in the last 50 to 60 years. Our population, because of our massive drug exposure, have just evolved some antimicrobial drug resistance factors that are just propagated in our population or in the population of normal flora. So this is actually kind of a generous look. There's probably a lot more drug resistance factors, but let's just say, going back to our example here, this is your gut. These are your normal flora. Now let's say that you get sick, right? You get sick with something, with anything, and you go and take a course of antibiotics. Let's say you got sick in your upper respiratory tract, right? You get a little lung infection. And sorry, that was not, let's say that you get a lung infection and you take antibiotics, you choose to take antibiotics, or specifically, right, antimicrobial drugs. And so now you put it into your body, it's targeting, you're trying to target that organ-specific disease, but of course it's gonna have impact on the normal flora too, so they'll be exposed to that drug. So even though your pathogen is up here and your antimicrobial drug is trying to get rid of this pathogen, that drug is now circulating in your body, especially if it's broad spectrum, it will then have a significant impact on the normal flora. So let's just say, again, this is still your normal flora. in the absence of pathogen, right? Your normal flora is going to be impacted as well with that broad spectrum medication. You will therefore, just within this population of bacteria in your gut, even though these are your normal flora, your good guys, quote unquote, you will be selecting for those organisms in your gut that have resistance to that particular drug. And so now you, right, let's say this is your exposure, and now you're healed. This is like your 7-day window. And now you're better, you're healthy again, convalescent. Which means that your infection is gone, right? You have no more infection. But what the impact of that drug was, not just cleared that pathogen, but now it also changed the gene pool in your gut. And so let's say that the second time around, or let's say that downstream, you accidentally inhale or eat a pathogen that comes down and hangs out in your flora, like salmonella, or let's say that you work in a hospital and you got exposed to Pseudomonas, or a really bad E. coli like EHEC, which is enterohemorrhagic E. coli, or ETEC or EPEC. Let's say that any of these gut pathogens comes into your body, they come down here, and now these pathogens, even if they're not drug resistant, they can now gain the resistance factor through conjugation, right, with these neighboring normal flora. And so they will then gain that drug resistance factor, and they will propagate because they're now a pathogen in your body. They're going to propagate, and each of these daughters are all going to now have that drug resistance factor. So even if you can get sick with a pathogen that is not drug resistant, if your gut looks like this, you may in the end shed drug resistant pathogens. Drug resistant pathogens. Are going to come out the other end, right? So the antibiotics, even though it can be beneficial, right, to clear an infection, be very aware, especially if you are taking a broad spectrum antibiotic or a patient is prescribed this, that it can have significant impacts on not just altering the normal flora, but in selecting for organisms that are best fit for that altered environment, so that the gene population or the gene pool in your normal flora is now shifted to have the majority of organisms in there, even the beneficial organisms, as having those drug resistance factors. And again, this is not going to hurt you, right? If it's your normal flora and they have drug resistance, I mean, maybe that's fine, because next time you take that same antimicrobial drug, at least you won't be impacting that same normal flora. But the biggest problem here is not with your normal flora, it's with pathogens that may come in and conjugate with your normal flora and therefore gain that drug resistance factor. And so now you're creating superbugs by essentially incubating them with this great antimicrobial resistance population in your gut. Of course, this can be true of pathogens too. So if there are pathogens that are drug resistant, and this is the population of pathogens in your gut or in your body during an infectious disease, you expose that pathogenic population, you still would be doing the same thing. The exposure to drug would select for organisms that are best adapted to that changing environment, same concept. And in this case, you would be keeping some level of that pathogenic population that now has drug resistance, and that could potentially propagate. Now, if you're otherwise healthy, then even though this might be the case, your body's immune system should be able to clear this infection before it has a chance to repopulate in your body. But the biggest problem with all of this stuff is not inside you, it's downstream. The next person who gets sick with this pathogen will now have a drug resistant pathogen that is no longer able to be targeted by that drug, right? This is just a general question. A man has a severe case of sinusitis that is negative for bacterial pathogens. A physician prescribes an oral antibacterial drug in treatment. What are your opinions of this therapy? can talk about this a little bit more in the lecture discussion, but I'm hoping that at this point you can stand up and shout loudly how irate you are at this prescription, because if you are not infected with a bacterial pathogen, there's no reason to take an antibacterial drug, right? No reason to. Yeah. Okay, so this slide, as mentioned, I've modified this a little bit. So because of this growing problem with antimicrobial resistance genes propagating in our population, not just of normal flora, but definitely of pathogens, and more we're hearing instances of people who are infected with a superbug, these multi-drug resistant pathogens, and it's becoming more and more challenging to find appropriate therapy to control these organisms. So pre and probiotics are one means. In fact, it's they're still not really permitted for therapeutic use, so you really cannot find any kind of probiotic drug, quote unquote, right, that is approved by the FDA to use against a bacterial or viral infection. But these certainly can bolster your flora inside your body. So prebiotics, if you recall this, these are nutrients that encourage the growth of beneficial microbes. These are things like, obviously, your fiber, galacto-oligosaccharide, inulin, right? All of these things that are pretty selective for the fermenting beneficial flora that are in your gut. And the fermentation of these prebiotics is not only beneficial for the bacteria, but also immensely beneficial for the human. So the prebiotics concept here is great if you have a decent flora in the first place, or at least as long as you're not missing those really beneficial microbes in your gut, then you can really encourage the growth of them and have overall health benefits to really help bolster your own immune response and, of course, your intestinal environment, et cetera, et cetera. Probiotics are the live bacteria. These ones do not colonize inside the gut. There's ample evidence that probiotics really don't stick around for longer than maybe a couple of weeks. They can, though, temporarily, while you are taking a course of probiotics, they will have actually quite significant impact on the environment of the gut. And if you're taking things like lactobacillus or bifidobacterium, these are both lactobacillus or lactobacilli are lactic acid fermenters. So they love to eat up the simple sugars in your diet, and in your small intestine, they'll produce a significant amount of lactic acid, which is good because you can drop the pH of your small intestine, which is where many pathogenic organisms like to go and bind to, like Salmonella and Shigella and E. coli. But they don't stick around for long periods of time. So in order to get the good effect of the probiotics, you need to have active, really daily intake of these live microbes. And in synergistic, in combination with these prebiotics, these things go together very well and can really alter the environment of the gut to discourage the growth of pathogenic organisms. Now, there are some people, and I won't ever say this as from a professional standpoint to recommend this, but I know anecdotally there are people that take pre and probiotics during the course of an infectious disease in hopes that it will actually overpower or change the environment significantly enough that the pathogen really loses its hold in that person. Again, there's no FDA-approved therapy for pre and probiotics yet for bacterial or viral infections, but this is definitely an approach that can be preventative, or downstream we may see that there is an application of this in treating infectious diseases. There's definitely experimental approaches where animals have been provided pro or pre or combination synergistic symbiotics to combat an infectious disease. And if you're ever interested, I can send you some articles on those because they're really fascinating studies, and there's a lot of power here, but just nothing that's approved yet. Bacteriophage therapy, I'm going to stand on my soapbox for a minute. This is something that has been around for actually centuries, almost, I can say at this point. So the Republic of Georgia, back when the Soviet Union was the USSR, the Republic of Georgia had an extensive library of bacteriophages, and we recall this from chapter 5. These are these little guys here. These bacteriophages are bacteria-specific viruses, right? And if you recall, again, the term specific is really important here. These phages are specific oftentimes down to the species or even subspecies level. And there are known bacteriophages for every single pathogen, every single bacteria, every single one. And the great thing about the concept of bacteriophages is that they adapt with their host, right? So if a bacteria develops resistance to a phage, then the phage will actually adapt and, or at least the phage population, not an individual one, but the population will adapt to that resistance factor and will also evolve too, right? So that they become infective again. So the concept behind bacteriophage therapy is that this is a pathogen targeted phage that a human can ingest or consume, right? So you have a pill or an injection, usually this is a pill that has these live virions, these bacteriophages, and if they're provided in a high enough concentration and they're provided in a targeted way, ideally, they will actually go and target that bacterial pathogen inside the human and get rid of that pathogenic population. It's a pretty fantastically phenomenal approach to antibacteria, right? Antibacterial therapy. This is not antiviral, it's antibacterial, so you can't use bacteriophages to eliminate a viral infection because we don't have any examples of viruses that infect viruses. But if you have a bacteria infection, then bacteriophage therapy is definitely one means by which to control for that population. Now, in the United States, we are not permitted to use bacteriophage therapy except for in compassionate care, which means you've exhausted all other possibilities and no antibiotics and no other therapy is effective against that particular pathogen, then you can change and do bacteriophage therapy. We actually have permission to do bacteriophage therapy in some agriculture. So actually in fish, in the aquatic industry, bacteriophage therapy is used in some circumstances more so than antibiotics to eliminate some very common bacterial infections in the fish population. So this slide here, and actually I have this on our Canvas page, a whole Word document that has more of these compare and contrast. But this is a super short and simple like bacteriophage versus antibiotics, and some of the benefit that you might find using a bacteriophage over a traditional antimicrobial medication. So here, the bacteriophages can be very specific, again, targeting species specificity and sometimes even subspecies, which is fantastic to eliminate that broad spectrum activity that we talked about before, right? Antibiotics targeting both pathogenic organisms and normal microflora. It's really hard to get a narrow spectrum antibiotic that is so targeted that it does not have impact on the normal flora. So of course, we've talked about some of these problems here with broad spectrum antibiotics or antibiotics that essentially overlap into the normal flora, versus bacteriophages are super narrow, and it's really uncommon to have any kind of downstream consequence to the specificity of bacteriophages. But of course, the disadvantage here is that you must have identified the etiological agent, just as we talked about before with narrow spectrum medication. The second one here, replicate at the site of infection and are thus available where they are most needed, right? So for phages, as long as you get a high enough concentration that you get them to the site of infection, you don't need to continuously administer these things, because once they arrive at the site of infection, they will infect the bacterial population and propagate on their own. So you don't need to continuously administer bacteriophages versus, of course, antibiotics. This has nothing to do with replication. So you have to have a continuous source of administration, which we've talked about before. Down here, no serious side effects have been described, right? I've mentioned this already. This was a technology, kind of a therapy that was developed many, many years ago in the Republic of Georgia. And unfortunately, when the USSR collapsed, then the Republic of Georgia essentially lost their facilities. They just had no more funding. They couldn't keep the lights on for quite a significant period of time, and really just lost their library. And because their language was not English, then most of the Western scientists did not pick this up. And it's taken a very long time for this to essentially be resurrected. There are a number of industries, bacteriophage companies around the world. We have, I think, 1 or 2 in the United States, 1 in Texas, I know. One of my friends works for one in Norway, and they're all, you know, getting ready to release their bacteriophage therapy library to the public, to the population, to human healthcare as soon as the FDA or, you know, the European FDA agency permits it. But for now, they still are really not permitted in our country, even though there really is no major detriment for bacteriophage therapy. So there are, of course, some side effects because, of course, if you have an active infection of an infectious agent, you're going to have lysed bacterial cells. Those lysed bacterial cells are definitely going to cause inflammation. So you certainly will have these side effects of inflammation at the site of infection, but you will already have that because you have an infected agent. So there can be some downstream consequences if there's release of endotoxins, for example, for EHEC or ETEC, something like that. But nothing like the significant side effects that antibiotics or antibacterial drugs can do, especially that toxicity stuff that we talked about before. So this last one here, CRISPR technology, I mentioned this one here, and let me just switch over to what this is. This is a gene targeting technology versus pathogen targeted phage. This one is actually targeting genes. And the general concept, CRISPR, by the way, the acronym here stands for clustered regularly interspaced short palindromic repeats. And you don't need to know what any of that stuff means, but essentially this is what's kind of cool is that this was discovered inside bacteria as a way for bacteria to gain immunity or resistance to viral pathogens. It's kind of cool. And unfortunately, this kind of is following our bacteriophage therapy conversation, but this is definitely one means by which bacteria can gain resistance to bacteriophages. But then I mentioned already that bacteriophages can adapt to even those resistance factors. But bacteria do it this way. They have this this really cool protein that's called the Cas9 system. And this is sort of this protein complex that has a guide RNA sequence in it. And this guide RNA sequence is directed towards a very specific gene sequence in a target DNA. Now, again, bacteria have developed this as resistance to viral invaders. So when a virus infects a bacterial cell, that viral nucleic acid is targeted. It's actually a piece of that fragment is taken out of that viral DNA and stuck into this CRISPR-Cas9 system. And so that the next time around, if that same population of bacteria gets infected with that same bacteriophage, then that little viral DNA segment or RNA segment that has been put into this little immunity system, this memory system, then that virus that comes in will have this CRISPR-Cas9 system immediately target that same gene segment on that viral DNA. And when that guide RNA matches up with a target DNA, then these enzymes, these endonuclease enzymes that are a part of the system will come and actually just cut out that DNA sequence. So this is what CRISPR technology is, or what the CRISPR system is about. This is a natural system that we have then taken out of the bacteria, and now we can use this for specific gene targeting of any cell, living or non-living. What the benefit of this is that it can be in living cells. And one of the applications of CRISPR is in antibacterial therapy, right? So if you have, again, a bacterial infection or even potentially a viral infection, you can take a pill that has these vectors, and we won't talk about that in detail, but a way to direct this CRISPR gene system into your infected, targeted to your infected tissue, but specifically that would enter into either the bacterial pathogens or into your infected tissue to provide your if this is a viral infection, then you'll provide your cells this CRISPR-Cas9 system that will target that pathogenic DNA, and it will actually just cut it up. It'll cut out whatever you're targeting. So if you target a metabolic enzyme, for example, or for a virus, if you target like the spike protein or the protein that's responsible for lysing that bacterial cell, or some key enzyme in the viral DNA or in the bacterial DNA, then you just essentially, you're just cutting up that one particular sequence, which will then cause that bacteria or virus to not be able to replicate anymore. So this definitely has some detrimental consequences. So there are some off-targeted gene deletions that can occur. So this is not yet approved for anything, right? For anything in the, actually that's not completely true. It's not approved for most things in the human population, except for in clinical trials right now for some experimental approaches to curing some single gene defects. For antibacterial therapy, this is definitely being explored, guaranteed, especially for those drug resistance pathogens that we have in the population and for viral infections. But as far as I know, we are far from getting approval for using the CRISPR technology for antibacterial, but don't be surprised if this becomes the next thing. All right, stepping back into our chapter. Sorry, my nose is very itchy today. So as mentioned, I mean, we talked about last week, we talked about with the last set of slides, the therapeutic index and the toxic dose, or TD50, right? That really with any type of chemotherapeutic agent that you put into the human body, even if it does not have direct toxicity to tissue, as you increase concentration, you'll eventually have some type of tissue toxicity. So we know already that there can be toxicity to tissue, either at low concentrations or at high, depending upon what the agent is that you're using. Some people have allergic reactions to the drug itself because the drug, as you administer it, it is a chemical that you're putting into the body, especially if you're doing this orally or intravenously, that it's getting into the blood and could invoke an immune system response, and specifically an allergic response, right? So if this does happen, then you can have things like anaphylactic response, which is essentially you stop breathing, your airways constrict, and everything can lead to your death. So it's very important if you or anyone that you know has any drug allergies, and especially for those of you entering into healthcare, that this be the very, very sensitive issue that you ask your patients about, and make sure that they've never had any type of negative or adverse response to any drugs in the past, and that you remember the drug classes and how similar one drug is to another, so that you can help reduce any type of potential allergic reaction in your patient. And then we've talked about this already, of course, that their broad spectrum antibiotics can certainly cause a disruption of the normal flora. This can lead to a superinfection of either your normal flora that have gained resistance and they can outgrow, or the more resistant flora that outgrow everything else. It can cause a secondary infection, as mentioned also before, with C. diff or Candida. It's really common. Secondary infections, or it can lead to general dysbiosis, which it almost always does. This is a picture of someone having gingival reactions, so they're having some type of allergic reaction to this drug, and it's becoming very evident in their gingival lining. All right, we are almost done. So things to consider when choosing a drug, and this is what we are going to practice in class with our in-class activities. I'll show you some case studies. So if you are interacting with a patient, a friend, yourself, right, and considering whether or not to seek antibacterial therapy, some things to consider when choosing the best type of antimicrobial medication or antibacterial drug, especially if you are in the healthcare industry, if you are a PA or you are actually going to go on to administer these medications, or even if you're not administering them, but you're listening in on conversations. These are things to consider when choosing the best drug. Number one, the nature of the microorganism. This is the pathogen that's causing the disease. We talked about this before, that broad spectrum medications are oftentimes prescribed initially or sometimes completely because of the limitation of requiring a diagnosis for providing narrow spectrum medication. So if at all possible, it would be great if we had a way to culture everyone's infectious disease so that we have the etiology, we know what's responsible for causing that infection. There are other ways that you can identify the agent, not just through patient samples, but through circumstances in the environment. We now have many very rapid immune-based tests that we could administer to patients to determine if they have viral versus bacterial infection or if they have a particular pathogen. So this is all about diagnosis. If possible, diagnose the infectious agent. Number two, before administering an antibiotic, as this is especially true now, I say that every year because we just have growing and growing and growing antimicrobial resistance factors in our population. And we have many, every year we're seeing more and more cases of people who are dying or who come near death because they have these drug resistance pathogens inside them. So this becomes a really pertinent issue that before you administer especially broad spectrum antibiotics, because those are the ones that are more those are the ones that have more antimicrobial resistance factors for them in our population, mostly just because we administer them so often. So before prescribing these things, you do need to, if at all possible, determine the degree of the microbe's susceptibility to drugs. Now in our class, we've already done an example of how you can do that. That's with the Kirby-Bauer disc diffusion assay. But I'll show you a slide in just a minute of how the clinical microbiology can accomplish this. So if you have a patient sample, oftentimes in addition to diagnosis, you can request a minimum inhibitory concentration assay where they essentially determine the concentration of many different drugs on the same pathogen to determine their susceptibility, not just whether they're resistant or not, but also the particular concentration at which that drug is effective or not effective. And then number 3, this is really just everything about the patient. So what are they presenting with, right? So the medical conditions, so what are they presenting with? What is their history? Do they have any contraindicated drugs that they're already taking? Any organ disease that might be inhibitory towards some of these drugs that you're administering, right? All of these things would go into that pool of patient condition or medical condition of the patient. So with the collection of all of these things, you should be able to then look at all of the potential antimicrobial medications we have available to us or to you and choose the best one for each of these different areas. As mentioned, we have this Kirby-Bauer test or agar disc diffusion assay that can be done to determine the microbe susceptibility. But again, this is not as commonly used in a clinical setting. This is more in a research setting, but in a clinical setting, we do something called the minimum inhibitory concentration. I've mentioned this in our last set of slides. This MIC number, the minimum inhibitory concentration, is the smallest effective dose of a drug on a microbe. And this is how you would complete this activity. Here is an example of something that you could do in the laboratory where you take some type of drug and you dilute it out. I'm sorry, I'm going backwards. So you take a drug here and you dilute it out. In this case, you can see it's a twofold dilution. So from 12 to 6 to 3 to 1, et cetera, et cetera. And then for each of these tubes that have a decreasing, we call it a serial dilution, right? So a 2-fold or a 10-fold factor of dilution of this drug. And you inoculate all of them with your bacteria and grow them, right? Grow them over a 12 to 24-hour period of time. And then you measure turbidity. And essentially what you're trying to find is the lowest concentration that you have inhibition of the bacterial population. It means nothing growing there. So in this example here, we would say 6.4 microliters would be, it should be like micrograms per microliter, would be the lowest concentration that is still inhibitory, because at 3.2 there becomes growth of bacteria, even if it's low, right? So the MIC would be 6.4 in this case. But just to show you a better example of what this actually looks like in the laboratory, instead of setting up test tubes for every organism for all the different drugs that you're testing, we have these 96 well plates. They look exactly like this. This is just cut off the bottom 2 rows. And these things can hold 200 microliters of your liquid. So you can actually test up to 7 different antibiotics at any given time. This is showing you increased or decreased concentration in this way. This is showing you decreased concentration in this way. Doesn't really matter how you, well, actually, no, that's the same thing. This is showing you increased concentration. Yeah, actually, these are exactly the same thing. So where you have the highest concentration on this end and you dilute out that particular antimicrobial agent over the span all the way down the side, and then you have a growth control inside. And this is just a diagram of what you might see, where you have growth indicated by that dark color, which would be turbidity, and no growth, of course, would be clear. And in this case, you can see that all of these compounds have a different level of effectiveness against the same pathogen, and you would just identify the lowest concentration that it is inhibitory as the minimum inhibitory concentration for that particular compound. What it would look like in reality is something like this, where you can see, hopefully, that these are all turbid samples here, and this is the first. Actually, this looks like this is the first clear sample. This is the first clear sample of this drug. This is the first clear sample of this one. And you can actually load this whole plate into a spectrophotometer much like the ones that we use to measure turbidity for bacterial growth back in the early part of our semester, except for it could read the entire plate at the same time, right? So a spectrophotometer could just analyze each of these wells and give you a reading so that you know where it's inhibitory and where you still have growth, and determine the MIC in a very, very rapid way. Of course, you have to let the bacteria grow first. But so this is how you would probably see this done in a clinical laboratory setting, definitely in a research setting, where you can analyze different the sensitivities of many different organisms at the same period of time. So these are some questions. They're not related to choosing the drug, but these are just kind of concept questions, really thinking about bacteriostatic versus bactericidal medication. So this first one, you've been directed to take a sample from a growth-free portion of the zone of inhibition in the Kirby-Bauer test and inoculate it onto a plate of non-selected media. What does it mean if growth occurs on the new plates, and what if no growth occurs? So again, just think about the concepts of bacteriostatic and bactericidal to answer this question. And the second one here is almost the same exact question. So think about this again, just thinking about bactericidal versus bacteriostatic, and see if you can answer this question. And then before I let you go, I'm gonna just bring this up for you in the classroom in our lecture discussion time. And in fact, this is how I like to ask you these concept questions on the exam as well. I'm going to provide you these three different kind of case studies where you can use that slide. Let me pull that slide back up again, just so I show you what you're doing here when you're doing this on your own time. This one. So use this slide, the one that says considerations when choosing the best antimicrobial drug. And when the question asks what considerations, or what are the considerations to keep in mind when you're choosing the best drug, you would literally state, number one, the nature of the microbe, and then go into the case information and see if you have information in the presenting case that can help you identify this organism. If you can't identify it right away, then you could say we would culture this microorganism. Number two, degree of microbe susceptibility to drugs. Again, go back to the case information and see if you can find information that might give you a clue as to whether this microbe is sensitive or not to particular drugs. Or just consider if this is a nosocomial-derived infectious agent, it is very likely that this is a drug-resistant pathogen, so you would indicate nosocomial patient or nosocomial infection, therefore likely drug resistance, so would request a minimum inhibitory concentration of different drugs, right, to determine susceptibility, or request a disc diffusion assay, whatever, something like that, to determine the susceptibility to different drugs. And then number three, the medical condition of the patient. And again, go into the case study information and glean out as much as you possibly can. So for example, this first case, I'm not going to do it with you here, but we'll do it in the lecture discussion time. In the post-op ward, your patient is a 47-year-old female recovering from a partial hysterectomy. This patient is on IV ciprofloxacin to control infection of the surgical site and is using a catheter while under mild sedation. On day 2 post-op, you become concerned when you see cloudy urine in the collection bag and request an immediate culture. Results come back positive for Pseudomonas, and the attending physician confirms a nosocomial-derived urinary tract infection. He discusses treatment options with you. So again, part A is what are the three considerations? Just go directly back to those slides, and it's going to be 3 different answers to this question, right? What is the infectious agent? Well, I've told you in this one already. What is the susceptibility to drugs? Go back through this case information, see if you can glean any of that information. I've already told you this is a nosocomial pathogen in this one. So again, think about that. And then what is the patient history? So you're gonna just pull as much of this information out as you can of this case study. And so with those 3 considerations, then you can go to your drug chart, your worksheet that you've been completing from your workbook for chapter 10, and look up all the drugs that we have available to us and figure out which one might be most appropriate for those conditions, right? So it requires a little bit of thought of the things you wrote down for your considerations that would allow you to help choose the best type of antimicrobial drug. And then just go and look up information about that drug. Are there major concerns or limitations to that particular antimicrobial medication that you choose? And then understand how it works, so how it targets that bacterial population. right? So that's how you're going to answer these questions. There's 3 of them, 3 examples I have here for you, and I think I might have another example in your study guide. So definitely go over those and be expected to answer these types of questions on the exam. Okay, that's it for this chapter and for these slides.